Okay, good evening, everyone. Can you all hear me? Yes. Okay. So we're going to continue with whatever we were seeing yesterday. What were we seeing yesterday? Okay, yeah. Agarose gel electrophoresis. So what happens here is we are separating the DNA fragments, right? So based on what? Based on two important features we are separating it. So whatever we saw yesterday is what I am telling you now. Okay. So based on what are we separating it if we see? So it is based on the size of fragment. Okay. So or in general the molecular weight of it, and then the charge of DNA which is negatively charged. Okay, so now where we are putting the DNA is on the side of the cathode. In the agarose gel that we have put, we have made these cavities which are called as wells, inside which we are loading our DNA sample. Okay, so DNA sample is loaded inside these wells, and then what we are doing, we are passing electricity through this. There is a buffer solution to conduct it through the agarose gel, and we also saw the agarose gel is going to be having a network of fibers, and this network is going to act as a filter. So the larger fragments of DNA, they stay close to the cathode, and the smaller fragments will move towards the anode. Okay. So now whichever size of DNA fragment that we have made, we know that is going to be our gene of interest. So now we are going to just cut this piece out, and we are going to take this DNA and dissolving out the agarose gel. We only take the DNA through a process that is called as elution. But then how you can visualize these DNA? You can't simply see it. It is all going to be transparent only. So that is why we are going to add a dye that is called as ethidium bromide. So what this ethidium bromide will do, it binds with the DNA, and then you are going to see these orange color bands of DNA, but again not under normal light, under the UV light. So when it is exposed to UV light, it is going to show these bands. So from this you will take the DNA strand. So this is the process of elution. Elution simply means extraction of this specific, let us say you want this specific DNA strand only. Okay, so that fragment you are going to take out of this agarose gel. So that process is called as elution, extraction of the desired gene from the agarose gel by removing the agar. So that is going to be the next step which we have seen, which is agarose gel electrophoresis. So far, if you remember, we were seeing about the different stages or processes in our DNA separation, making our rDNA. So in that, the first step was isolation of DNA from the donor cell, which we have done by using just the enzymes. And then we also saw about fragmentation of DNA, because you don't need the whole DNA, you just need your gene of interest. So you are using suitable restriction enzymes, and these restriction enzymes will cut only specific pieces of gene of interest. And that gene of interest, how we are going to separate out of all the fragments that you are having, for that we have used your agarose gel electrophoresis. And then next, breaking up. What I will do now? Wait just 1 minute, ma. Is it clearly audible now? Yes? Okay. So if it breaks again, please let me know, ma. Okay? Right. Okay. So now we are moving on to the next part. So now so far what we have done, we have isolated the DNA. Isolation was done, and then now you have your gene of interest. But how many copies you have? We have very few copies, or even sometimes only one copy only you are going to have. So now the next step, the step number 4, is amplification of DNA. So how we are going to amplify the DNA? This is through the process called PCR. So this is the polymerase chain reaction. So polymerase chain reaction is again another important process. Now once we are done with this polymerase chain reaction, the lesson is as good as over. because after this everything is going to be quite simple. So I'm assuming you have studied about PCR in botany in molecular basis. Yes, have you studied or not? Am I audible? Okay, have you studied about PCR? Just say yes or no. No. Okay, fine. Okay, so see, PCR polymerase chain reaction is going to be helping in amplification of DNA. I think yesterday in class I mentioned in very short span you will get 1 million DNA copies, and if you repeat this process, let us say for 30 cycles, you can get even 1 billion copies of your gene of interest. So how this process goes on is what we are going to see. So there are 3 stages in PCR. Okay, so now first this method, this process, it was introduced by or devised by a person named Carey Mullis, and the a name you have to remember. So Carey Mullis devised this polymerase chain reaction. So basically we are going to make copies of DNA by 3 major steps. So there are 3 steps that are involved in this amplification of DNA. First is called as denaturation. The second step would be annealing, and the third step, extension. So denaturation, annealing and extension, these are the 3 steps that are involved in amplification of DNA or PCR. Okay, so before that you should know this PCR is not just a process, it is a machine also. Everything happens inside this machine. It is going to control the environment in which the reaction is going to occur. So PCR device is going to be a machine inside which all of these processes are happening. So first we'll see what exactly goes on in this process, and then we'll see what the machine looks like. Okay, first denaturation. Now here the important thing for you to remember is we are trying to amplify your desirable gene. Don't write, please listen only. Okay, anyway, because I'm going to post the notes and I'll post the video also, so please first listen. Okay, so desirable gene you have isolated now by doing 3 major steps so far. Okay, so now this is going to be your desirable gene. So it will have 3 prime to 5 prime, 5 to 3, or the other way around. Okay, so now this is going to be your gene of interest. Okay, so now you have to amplify it, and the first step here for amplification is a process called as denaturation. So this is going to be what we call as the template strand or the parent strand. So we need to make copies of this template strand only. So what we are going to do is we are going to denature it. So how we are denaturing it is majorly everything in this is going to be happening through temperature. So the first step, denaturation. So what happens in denaturation is simply we are increasing the temperature so that the hydrogen bonds are broken. So in denaturation there is going to be breakage of hydrogen bonds. So if hydrogen bonds are broken, what happens to the template strand? What happens to the template strand if the hydrogen bonds are broken? Someone please tell me. Huh? Okay, yes. So now we are going to get 2 single strands as a result of this. Okay, so this 5 prime to 3 prime. Okay, so this is going to be one strand because what happens here is that the 2 strands are held together by hydrogen bond only, which now breaks. So now this process is called as denaturation, and denaturation occurs at what temperature is? It occurs at around 94 degrees Celsius. Okay, So that is denaturation. So there are now two parent strands that are forming. So it is occurring at 94 degrees Celsius, 94 to 95 degrees Celsius, and this takes only 2 minutes. Okay, so this is going to be happening in just 2 minutes. You are going to get these two strands. So this is the first step, denaturation, breakage of hydrogen bonds, where the DNA is going to break into single strands. This is the first step. Please follow carefully. If you're not hearing me or if you can't see the screen, please let me know. Okay. Right. See, denaturation is done. So next step is called annealing. Now first step was very simple. Now the second step, annealing, is attachment of primers. This I think you know what is a primer. Can someone tell me what's a primer? What is a primer? How? What is a primer then? What is it made up of? RNA? Okay. Why is we are using an RNA and a DNA? Yes? Yes, Anto. Anto, tell. Give me the answer. How? Why? Why? What is the connection? Faster replication. Okay. Do you know why is it called a? In which topic did you study this? Okay. Okay. So then, if a DNA strand is going to have an RNA, how would that? If it is having an uracil, then how does it become a DNA? How is it a DNA? Okay, anyway, we'll see. So attachment of primer. Now, what is this primer is? This is going to be a single strand of DNA and it is an oligonucleotide. So what do we mean by this oligonucleotide? It is going to be a short nucleotide sequence. So this is simply a very short nucleotide sequence. So that is what an oligonucleotide is. And this short nucleotide sequence will be complementary to the template strand. So complementary to the template strand. So this is going to be annealing. Okay. So now basically let us say this is going to be, don't draw, write anything. Hope you are all listening. So let us say this is going to be one strand. So what the primer is going to be? Something. So this is going to be my strand. So what will be my primer is, see, primer, I'm assuming you all know that it should always be attached to the 3' end of the template strand. So it is going to be an oligonucleotide, meaning it is going to be a short nucleotide sequence only. So here what would be the primer that I'm using? What would be the sequence? I say, hmm, hmm. CTAG. Okay, so this is going to be CTAG. This is the primer that we are using. So that is all a primer is. See, why a primer is used? The function of a primer is to initiate what? Initiate what? Yes, it is to initiate replication. So this is going to start the process of replication. So that is what we are doing. So this first stage here is going to be denaturation, where we are separating the strands. In the next step, which is called as annealing, what we are going to do is we are going to attach. Your chemistry sir is disturbing the class.
Got that recording.
Yeah, I'm recording only. I know, after you went it started recording. All this is also recorded, Vani sir. 5 prime to 3 prime and then 3 prime to 5 prime we have. So here what we are going to do is we are going to add a primer to one end. Always attached to the 3 prime end of the strand. So this is to initiate the process of replication. So how many primers are needed here? Definitely how many primers are needed? Since there are two strands, two primers, but in our NCRT it is given as two sets of primers, so that is what we are going to see. So there is going to be two sets of primers used, okay, which is going to be definitely complementary to which strand? To the template strand. So that is what we are seeing here. So this is attachment of primer. So that second stage is going to be referred to as annealing. So denaturation occurred at 94 degrees Celsius. So annealing should occur at what temperature? Should it be higher than 94 or lesser than that? Lesser than that, because if it is higher it is going to break the hydrogen bond, that much we know. So if you are again putting it through higher temperature, it will break again, so we don't want that. So this is going to happen at a lesser temperature of around 50 to 60 degrees Celsius. So this process is called as annealing. Okay, so now annealing is done. If you have any doubts in this, please ask fast. Yeah. And that is what I said. That is what I'm saying. That is what I'm saying. We are seeing whatever is there in NCRT. Okay. Very last year NEET question paper. This was one of the questions that had issues. Two sets of primers or true primers. So in NCRT it's clearly given two sets. So it's two sets of primers. Okay. Okay. So next step is extension. Now this is where we are going to use some very important things, something that the name has. So extension, how? How? With the help of what? Very good. Yes, so extension of the primers with the help of polymerase. And do you remember we saw about a specific type of polymerase? Very good. Yes, so it is stack polymerase. So it is stack polymerase. So this stack, I hope you all remember, it stands for thermus. Yes, thermus aquaticus. Okay, so why we are using Thermus aquaticus polymerase? Why can't we use any other type of polymerase? Is because this process of extension it occurs at 72 degrees Celsius. So which means, can a normal polymerase or your own polymerase, can it withstand its temperature? No, it cannot withstand this temperature. So as a result, what will happen? It will denature. So to prevent that from happening, they are using Thermus aquaticus, which is going to be what type of bacteria? Yes, bacteria which is found in hot water springs. So this is going to be an thermoacidophile. So now here the third stage is going to yes, yes. If the lesson is not getting over today, it is purely because of your chemistry teacher. Okay. No, sir. Thank you, sir. No, no, sir. Please, sir. You completed questions for the 12th questions over. Okay, so the next step, I'm very sorry for the interruption by your chemistry sir. Okay, so now here we have already added the primers. So extension simply means what? More nucleotides are going to be added, okay, complementary to the strand. So now what has happened, in the place where we had only one strand initially. Now we have formed two strands. So now two strands are going to be formed, where the parent strand is going to be a part of it. The parent strand or the template strand is definitely going to be a part of it, but then now what we also have is a new strand that has formed. So this is what happens in case of polymerase chain reaction. Okay, so three stages, where in the first stage what is happening, please listen. So in the first stage, so this is going to be our template strand. So in the first stage there is going to be denaturation. So in the second stage there is going to be annealing, which is attachment of primers, and the third stage is going to be extension, where the primers are simply going to be extended, and we are going to get now two strands where initially there was only one strand present. Okay, so this is going to be how through every cycle we are going to multiply our primers, multiply our template strand, which is basically our gene of interest. Did you all understand what's going on here? Okay, so now this is what happens. So now again in the next stage what you will have, you are going to continue this process. So there are going to be again two strands that are present, and then again two more strands are going to be formed. So now two strands have become four, and it is going to continue like this until the cycles are over. So if we keep doing this for around 30 cycles, the copies that you are going to get approximately is 1 billion copies of DNA. So 1 billion copies we are going to get by 30 cycles. So this is so that you can multiply your gene of interest. Did you all understand what is going on? If you have any questions now, please ask. Raghu, can you please? Uh, where did you see it? Where? In what context it is given, ma? Should be a recombinant DNA only, because see, normally what a chimera is, an animal, I think you all know, a mythological animal which is going to be having multiple animals. It has a lion head, snake as a tail and all that. Chimera usually means combination of something. So I'm assuming it should be an rDNA only. Can you check the answer? Please check, Raghunandan, and tell me what answer they have given. That is rDNA, right? Joining the alien DNA. So if there is a plasmid and if you are joining an alien DNA, that is our rDNA, right? Yes. Hey Harris, you have sent something just now. I'm seeing primer has only four. No, that depends on the length of gene and everything. Taq polymerase helps in extension of, yes, Harris. Taq polymerase helps in extension of primers. Okay, fine. So this is what happens with PCR. So where what happens? Just we are going to amplify the gene of interest that we have. So gene of interest amplification is done. So now you have multiple copies of your gene of interest. What is the next step? What are we going to do next? What do we do next?